Ever seen that English ivy just relentlessly climbing up a wall? It looks so passive, but under the surface, it's like an engineering marvel going on. All that water and nutrients moving up against gravity. It really makes you appreciate how hard it must have been for the first plants coming out of the water onto land.
Oh, absolutely. Like for algae, it was simple. Everything they needed was just right there. Water, minerals, CO2. Totally available. But on land, it's a whole different ballgame. Finding water when you're not swimming in it, getting CO2 from the air without drying out, staying upright. Plants had to come up with some seriously creative solutions.
And that's exactly what we're diving into today. The amazing mechanics of how vascular plants get and move what they need. We've got this chapter all about it, and we're going deep, like we're going to understand it all.
It's really fascinating stuff. And as we go through it, you'll see how connected everything is. The way plants adapted, their roots digging down, shoots reaching up, leaves grabbing sunlight. Then we'll trace how those resources actually move, both short distances within cells and tissues, and long hauls through the whole plant.
So we'll be tracking that incredible journey of water and minerals pulled from the soil way up to the top, all driven by transpiration. And we'll see how they manage that water loss through those tiny stomata. It's a real balancing act, taking in CO2 for photosynthesis and not turning into a crisp.
And then, of course, we have the sugars, the fuel from photosynthesis, how they're transported right where they're needed. Plus, we'll touch on the symplast, this interconnected network inside the plant, which is like a super highway of communication.
By the end, you'll have a real grasp of how vascular plants work, this whole system of taking in and moving stuff around. And I have to say, some of this is pretty mind-blowing. You know how much water a corn plant moves in a season? It's a crazy amount. Or the pressure it takes to pull water up a redwood. Unbelievable.
Oh, yeah.
So back to the beginning. Plants conquering land.
Yeah, going from that all-you-can-eat buffet in the water to having to hunt down every single ingredient. Those early land plants, the non-vascular ones, they had to stick to damp spots. Developed a waxy cuticle and those stomata. It was all about water conservation, but still allowing enough gas exchange. And to anchor themselves. Simple stuff. A modified stem base or rhizoids.
Nothing like the elaborate root and shoot systems we see today.
Nope. The game changer was vascular tissue, xylem and phloem. That's what made long-distance transport possible, which then allowed roots and shoots to evolve as we know them. Xylem carrying water and minerals up, phloem moving sugars from the leaves to the rest of the plant.
Okay, so now with the plumbing in place, plants could branch out literally.
Yeah.
Speaking of branching, why do some trees like palms have a single trunk while others are super bushy?
It's a resource allocation game. A plant has so much energy to spend on growing. Branch out a lot, maybe not as tall, could get shaded. Focus on height, might miss out on capturing sunlight. It's a balancing act, influenced by genes and the environment.
And leaf size. Tiny pygmy-weed leaves to those giant Raphiregalis palm fronds. Makes sense that water availability plays a huge role.
Absolutely. Big leaves are great for soaking up sunlight and CO2, but they lose more water too. It's that trade-off again. Photosynthesis versus water loss. Dry climates. Small leaves to minimize surface area.
And phyllotaxy. The arrangement of leaves on a stem. Sounds technical, but it's about optimizing that light capture too.
It is. That spiral pattern, about 137.5 degrees between leaves, is common and efficient at minimizing shading. Nature's geometry at work. But in super sunny spots, opposite leaves might be better for some self-shading to prevent overheating.
And when those lower leaves aren't getting enough light, that's when self-pruning happens.
Exactly. If they're using more energy than they make, they're a drain. So the plant cuts its losses, redirects resources to the productive parts.
We also have the leaf area index. Seems like more leaves would always equal more photosynthesis, but there's a limit.
There is. Too many leaves, and the lower ones get too shaded, diminishing returns. You even see self-pruning happening then. Knowing the ideal leaf area index for a crop is super important for farmers.
And even the angle of the leaves matters. Flat to catch low light, vertical to avoid scorching sun, and let light deeper into the canopy. So many strategies.
Amazing, isn't it? And just like the shoots are optimized for light, roots are all about water and nutrients from the soil. They're not just passively absorbing. They change their architecture based on what's available.
Like those roots that branch like crazy where there's nitrate, but grow sparsely where it's scarce. It's like they're hunting for it.
They are. They sense nutrient levels and adjust their growth. Even the root cells change, producing more of the right transport proteins. Efficiency in action.
And that buffalo grass example. Fewer roots when planted near their own cuttings compared to unrelated ones.
Yeah.
Reducing competition within their own family.
points to some level of self-recognition, which is a fascinating area of research. It's about maximizing resource use as a whole.
And of course, we can't forget mycorrhizae. Those fungal partnerships are essential.
Absolutely crucial for water and especially phosphate uptake. It's a total win-win. Plants get more resources, fungi get sugars. We'll be doing a whole deep dive on mycorrhizae later on. They're that cool.
Okay, so plants have gotten what they need. Now, how do they move it around? Short distances, long distances. It seems like there are different routes for everything.
You've got it. And we have two main compartments to think about, the apoplast and the symplast. Apoplast is everything outside the cells, cell walls, spaces between them, even the insides of dead xylem cells. Symplast is the network of living cell interiors, all connected by plasmodesmata.
So apoplast, like an external highway, symplast is internal, all connected.
A good way to think about it? And there are three routes for water in solutes. Apoplastic through that extracellular space, symplastic crossing one membrane and then moving through the connected cytoplasm, and transmembrane crossing multiple membranes as they go. And it's not always one or the other. They can use a combo depending on what's being moved.
For short distances across cell membranes, plants do things differently with those hydrogen ion pumps.
Yep. Not sodium-potassium like in animals. Plant cells use proton pumps. They burn ATP to move hydrogen ions out, creating a concentration gradient and an electrical charge difference. That gradient then powers the movement of other things.
Like sugars, sucrose, and nitrate, getting in by hitching a ride with the hydrogen ions flowing back in.
Exactly. Moving against their concentration gradient, it's secondary active transport, using that stored energy. And like animal cells, they have ion channels too, those selective pores for rapid ion movement. Potassium channels are key in how the stomata open and close.
And we notice plant electrical signals are slower than animal ones, often using calcium instead of sodium. What's the reason for that?
Great question. Animals need super fast responses for movement and reacting to danger. Plants, they're generally staying put. They need to sense and respond to, but not at lightning speed. Slow and steady, that calcium-based system works for them.
Okay, on to short distance water transport, osmosis, and water potential. I have to admit, all those side symbols are a bit intimidating.
It looks complex, but the idea is simple. Water goes where water potential is lower. Think of it as water's potential energy compared to pure water. Two factors matter: solute concentration and pressure.
So more solutes mean lower water potential because they bind water molecules, less free water to move. And pressure can add or subtract depending if it's pushing or pulling.
Exactly. Solutes always lower it. Pressure can be positive, like turgor pressure inside cells keeping the plant stiff, or negative, like tension pulling water up the xylem. Water wants to even things out.
That figure with the flaccid, plasmolized, and turgid cells in different solutions was super helpful. It really showed how water potential works.
A classic demonstration. Flaccid cell in a solution with lower water potential. Water leaves, it shrinks, that's plasmolysis. Put it in pure water, water rushes in, it swells, that's turgor pressure. Vital for structure.
And then we have aquaporins, those specialized channels speeding up water movement through the membrane. And their activity can be regulated too.
That's right. Water can diffuse on its own, but aquaporins make it much faster, like dedicated lanes on a highway. And the plant can control how permeable they are using calcium and pH, fine-tuning the flow.
But for water to get from roots to the top of a tree, we need something faster than diffusion, bulk flow.
Exactly. Diffusion is fine for short distances, but too slow for the whole plant. Bulk flow is like fluid moving through pipes, high pressure to low pressure. It carries everything along.
And this is happening in xylem and phloem, right? Their structure is perfect for it.
Yep. Mature xylem cells are dead, basically hollow tubes, and they have perforation plates and pits to help flow and prevent blockage. Sieve tube elements in phloem, they're alive, but with a minimal cytoplasm and sieve plates with big pores for the sugar-rich sap. The veins ensure every cell is connected to this highway system.
So the plant has its own plumbing, and the speed difference is incredible compared to diffusion.
It is. Meters per hour in xylem versus years for diffusion over the same distance. Pressure makes that rapid transport possible.
Which leads to the big question. How does water defy gravity and climb up those trees? No heart to pump it. Transpiration seems to be the key.
That's the core of the cohesion tension theory. When water evaporates from leaves, it creates tension. Water molecules stick together, cohesion. They also stick to the xylem walls, adhesion. It's like a continuous chain.
So that tension pulls on the water below, And because it's all linked, the pull goes all the way down to the roots.
Exactly. And adhesion helps counteract gravity, keeping that chain intact. Water under tension in xylem is surprisingly strong.
And it's all powered by the sun driving evaporation. The plant isn't spending energy to lift the water itself.
A crucial point. Xylem sap ascent is mostly passive. The plant does spend energy absorbing minerals in the roots, which influences water uptake, but the upward movement itself is free.
But what about those super tall trees? What if that chain breaks? We talked about cavitation, those air bubbles forming.
It's a risk, especially in wider vessels. Drought stress or freezing and thawing can cause it. Those bubbles can block the flow. But plants have ways to minimize and even repair it. Alternative paths through pits, root pressure pushing bubbles out. In trees, only the newest xylem is active anyway, so there's backup.
To sum up xylem transport, bulk flow, driven by a water potential gradient, mainly the tension from transpiration, happens in those dead, hollow cells, moving everything together much faster than diffusion ever could. Super efficient.
It is, and that pull goes all the way down, creating the gradient for water to move passively from soil to roots to shoots.
And transpiration is essential,
Hmm.
but it also means losing water. How do they control that? Stomata and guard cells.
Exactly. Leaves need to be big for photosynthesis, but that means lots of potential for water loss. About 95% of it happens through stomata, even though they're a small part of the leaf. The cuticle helps in the other areas.
And guard cells are the gatekeepers, changing shape to open or close the stomata, balancing CO2 intake with water conservation.
Right. Turgid guard cells pore open. Flaccid, it closes. And it's mostly potassium ions driving that change.
So how do they know when to open or close? It's got to be more than random.
It's highly regulated. Light, especially blue light, triggers opening at dawn. Low CO2 inside the leaf also signals opening. And plants have an internal clock, the circadian rhythm, that helps too.
And under drought stress, they produce abscisic acid, ABA, which tells the guard cells to close even if there's light. Water conservation first.
They're constantly integrating all these signals, light, CO2, water status, even temperature. It's dynamic control for that photosynthesis water loss balance.
And transpiration actually cools the leaves down like sweating.
It does. Water evaporating takes a lot of heat energy, important for preventing damage in hot conditions.
Then we have xerophytes, those desert specialists. They have a whole arsenal of adaptations.
Incredible examples of evolution. Thick cuticles, sunken stomata, reduced leaves. CAM photosynthesis for nighttime CO2 uptake. They've adapted their structure and function to thrive in those dry conditions.
Okay, water's journey is complete. Now on to the sugars made during photosynthesis. They need to get around the plant too. Translocation in the phloem. And it works differently than xylem.
It does. Xylem flow is mostly one way, up. Phloem sap, that concentrated sugar solution, can go in any direction, from source to sink.
Source being where sugar is made, like leaves or storage organs when they're breaking down starch. Sink being where it's needed or stored. Roots, buds, fruits, growing leaves.
Right. And sinks usually get their sugar from the closest source, so direction isn't fixed. It depends on where the sugar is needed.
So how does sugar get into the phloem in the first place? Phloem loading. And it seems there are different ways depending on the plant.
There are. Sometimes it's mostly through the symplast, those plasmodesmata connections. Sometimes it's a mix of symplastic and apoplastic, and often it needs active transport to get that sugar into the phloem.
And that's where proton pumps come in again.
Exactly. They create a gradient that's used to co-transport sucrose across the membrane. Same chemiosmotic mechanism. Some companion cells even have special wall ingrowths to increase surface area for more transport.
So once the sugar is loaded, how does it get to the sink? The pressure flow hypothesis, right?
That's the leading explanation. High sugar concentration at the source lowers water potential. Drawing water in from the xylem increases pressure. At the sink, sugar is used or stored, keeping concentration low. Water leaves, pressure drops. That pressure difference drives the flow.
Like squeezing toothpaste. High pressure pushes it towards the open end. And at the sink, sugar is constantly being used or stored, maintaining that gradient. The water then cycles back to the source through the xylem.
Exactly. An elegant system using pressure for long-distance sugar transport. But in non-flowering plants like gymnosperms and ferns, it might work a bit differently. There's still debate on the details.
So what are some of those alternative hypotheses?
Well, some researchers propose that in these plants, there might be a contribution from cytoplasmic streaming within the sieve tube elements. That's the active movement of cytoplasm driven by contractile proteins. It could potentially help to move phloem sap along, especially over shorter distances. Others suggest that electroosmosis, the movement of fluid driven by an electrical potential, might play a role, but more research is needed to fully understand phloem transport in these lineages.
It always comes back to more research, but that's what makes it so exciting, right? There's always more to discover.
Well,
And we haven't even touched on the dynamic symplast, this incredible network of interconnected living cells. It's much more than just a transport system.
Oh, absolutely. It's like the plant's internal communication network, information superhighway. And those plasmodesmata, those tiny channels connecting the cells, they're not just static tunnels. They can change their size and permeability, even close completely, in response to various signals.
So it's not just about moving sugars and water around. It's about coordinating the plant's activities, responding to changes, even defending against pathogens.
Precisely. There's a whole level of regulation happening at the plasmodesmata level, controlling what moves between cells and when. And it can be very specific, allowing some molecules to pass, but blocking others. And this regulation can change over time, too. For example, as a leaf matures and transitions from a sink to a source, the plasmosmata connecting it to the phloem can become more restrictive, preventing sugar from flowing back out.
It's like the plant is constantly remodeling its internal connections. And the fact that viruses can exploit this symplastic network to spread throughout the plant is both fascinating and a little terrifying. It highlights how interconnected everything is.
It does, and the way some viruses actually modify the plasmodesmata to make them wider, allowing their larger viral particles to pass through, is a remarkable example of coevolution.
Now, we briefly mentioned electrical signaling in plants earlier, but it seems the phloem plays a role here too, not just those slower calcium-based signals.
It does. In some plants, like the sensitive plant Mimosa pudica, which famously folds its leaves when touched, rapid, long-distance electrical signals can zip through the phloem, triggering those dramatic responses. It's a bit like a nerve impulse in animals. And there's growing evidence that these electrical signals might be more widespread in plants than we initially thought, potentially involved in coordinating responses to stress, regulating growth, even influencing flowering time.
So it's not just about the physical movement of resources. There's this whole level of information exchange happening too, coordinating the plant's responses to its environment and its own internal needs. And all of this incredibly complex system of acquisition, transport, and communication, has evolved to solve one fundamental challenge: getting the resources a plant needs from where they are to where they need to be.
It's a beautiful example of how evolution can lead to these incredibly elegant and efficient solutions. And it highlights how plants, despite their seeming stillness, are actually dynamic and responsive organisms, constantly adjusting and adapting to thrive in a world of ever-changing conditions.
And it makes you think about how environmental changes, like drought or increasing temperatures, might impact these finely tuned systems. How will plants adapt to the challenges of a changing world?
That's a question a lot of researchers are grappling with right now. Understanding these fundamental transport mechanisms is crucial if we want to predict how plants will respond to future climates and potentially help them adapt. It's also relevant for agriculture, of course. Optimizing these processes is key to maximizing crop yields and ensuring food security for a growing population.
So much to think about. And it all started with that simple image of ivy climbing a wall. Who knew there was so much going on beneath the surface? Thanks for joining us on this deep dive. It's been an amazing journey.
It has, and I hope it inspires you to look at plants with a newfound appreciation for the incredible things they're doing all around us every day.
And to think about the intricate connections between plants and their environment, and how those connections might be changing in the future. What new adaptations might we see? What innovative solutions might they evolve? It's a question worth pondering.
It certainly is. And it reminds us that even in the seemingly still world of plants, there's constant motion, adaptation, and innovation happening all around us.
Thanks for listening.